invite Dr. Juby ma'am from an JSS College of Pharmacy, and it's my pleasure to introduce her in this warm evening to everyone. She is Dr. Juby ma'am is an associate professor and Department of Chemistry, Pharmaceutical Chemistry, JSS College of Pharmacy, OT, and she has been a faculty member of JSS College since 2007. She earned her B Pharm in SB College of Pharmacy, and M Pharm in KM College of Pharmacy, and PhD from a JSS Academic Institute of Education Research, Mysuru. And she has been worked on various research activity, including in-silico, in vitro, and including specialization in drug and scaffold repurposing. And she is one of the best outgoing students during her M.Pharm studies. According to her accolades, she has published more than 50 international papers and 29 national papers in a highly indexed Scopus and Sky Index, and she is having 15 H-index journals. And moreover, she has 2 Indian patents. She also completed her research grant sponsored by DST, and she also received a travel grants from DST and ICMR. And moreover, I can add her, and she is a recipient for an Young Research Award of 2019 and Young Performer of the Year 2020 from an Associate of Pharmacy Professionals India. With this brief profile, I am very happy and delighted to welcome Dr. S. Juby ma'am for this great evening to deliver a talk on about the effect of polyphenols and multi-targets of SARS-CoV-2 as a kind of a systematic in silico study. Over to you, ma'am.
Thank you. Thank you, Divya ma'am, for your nice. Yeah, ma'am. Thank you, ma'am. Is my screen visible?
Yes, ma'am. Your screen is visible.
And okay. Thank you so much, Divya ma'am, for this nice opportunity. First of all, my thanks to Altem Technologies Limited for sharing my knowledge on this particular topic. In fact, this work has been supported by them. They have given the COVID evaluation license for our short-term study. So with this brief introduction, let us start the study. So my title is Effect of Polyphenols on Multiple Targets of SARS-CoV-2, a systematic in silico study. So what are the highlights of our study? So as we all know very clear, this coronavirus disease, that is COVID-19, is a zoonotic disease. It's caused by a severe acute respiratory syndrome, which has quickly resulted in a pandemic. So maybe in the last of 2020, we aimed to identify the phytochemicals which have potentials to inhibit SARS-CoV-2 infection via multiple targets. So for that, we have selected 132 phytochemicals of various chemical nature, like phytotannins, alkaloids, carbohydrates, but which have been already reported for SARS-CoV and MERS, all these coronavirus related family diseases. So first of all, we started with structure-based in silico technique, again using main protease MPRO. Then after that, we narrowed down our results to CDocker module of Discovery Studio. Then we have selected 6 analogs of gallocatechin gallate. Okay. Then further we confirmed these studies with molecular dynamics studies. Okay, so this is all our highlights of the study. So the whole study only we are going to discuss today. Yeah, so this is about the coronavirus. So it is a family of viruses affecting the respiratory tract. So it normally causes the symptoms. It starts from common cold to pneumonia. So usually this virus, it lives in bats and other wild animals, and it is transmitted to humans directly or via other animals. It can also transmit between humans via respiratory droplets. Okay. So we know very clear this infectious disease is responsible for this 2019-20 viral pneumonia outbreaks. So though we have so many prophylactic vaccines in our hand, this disease, it's very difficult and very difficult to treat, and still we are suffering from this disease. So there are so many initiatives like US FDA has started CTAP, that is Coronavirus Treatment Acceleration Program, which is to initiate the COVID-19 clinical trials, and so many drugs have been given for the treatment like chloroquine, hydroxychloroquine, remdesivir, but still it is a challenge for us. So that's what is our thought process. Why not we go for multiple targets rather than targeting a single enzyme? Why not we go for a multiple targets, so we can forego various issues like resistance issues, all those things we have thought process. Okay, yeah, so this is the structure of the coronavirus. So it is a member of family Coronaviridae, and it is a single stranded RNA virus, we all know very clear, and the size is 80 to 220 nanometer, and it is a distinct and large club shaped spike protein. So normally this S protein, it binds to ACE2 receptors, especially in the lung and nasal epithelial cells. Okay, so here this transmembrane serine protease 2 primes S protein, it facilitates the entry of the virus into the host. Yeah, so this ACE2 and TMPRS2 are enriched in nasal tissues and nasopharyngeal passages. And these are all the variants of this SARS-CoV-2, starting with alpha, beta, gamma, delta, and now we have the Omicron variants. Okay, and these are all the countries which have first noted this particular variant and when it has been designated the particular variant. Yeah, and coming about the infectivity and mortality, these are all also the facts we know all very clear. So it is a jump species and more deadly, highly transmissible, increases vulnerability to infection and disease. So among alpha, gamma, and delta variant, the various data suggest that the Omicron variant may be more transmissible than delta and it may spread faster. Overall, it has a high mortality rate, and the IFR, that is infection fatality ratio, it differs 1.15 percentage in high income nation versus 0.23 percentage in low income nation. So the probability of fatal disease is highest in aged population more than 65 years, and the symptomatic infection prevalence more than 45 percentage, and the asymptomatic patients can have 75 percentage of transmissibility. More than 7 million infections and more than 5 million deaths worldwide since the pandemic began. And coming to the clinical features, so normally the older males more than 50 years, particularly those with underlying comorbidities, they are more prone for this COVID-19, especially those who are having the comorbidities like obesity, high blood pressure, cholesterol, diabetes, heart and kidney diseases are mostly likely to affect by this disease. So the common clinical manifestations of illness, it starts with fever, chill, cough, fatigue, muscle, body aches, loss of taste and smell, shortness of breath, nausea and vomiting. And it develops the ARDS also, that is acute respiratory distress syndrome. And not only that, it also like some pro-inflammatory cytokines like IL-1 beta, especially this IL-6, interleukin 6, TN alpha and IL-10, they also will be increased in these severe cases. Yeah, so this is the life cycle of coronavirus. So it starts with the entry the spike proteins, they will enter into the a host cell membrane via ACE2 receptor. So here this TMPRSS, that is transmembrane serine membrane protease 2, it will modulate, or it will help the spike proteins to get introduced into the host. Once the virus is entered into the host, it goes for uncoating, and the genome or the nuclei part, it will be entered into the host, the RNA. Here this RNA, it will be cleaved, the polypeptides will be cleaved into small proteins. So here only the major protein, that is main protease, or the main protease, it plays the major role. After that, it will be the RNA polymerase will come into the picture, that is the synthesis of the RNA. So once the RNA has been formed, and all the structural proteins got into assembly, and it will be released by budding process. Okay, so here you can see we have given some of the targets. Here only we started our targets. So first target is this ACE2 receptor. Okay, so why not the compounds? Why not the particular compound? It may be synthetic, or it may be the natural compound. Why not it inhibit the ACE2? That is the entry level itself, the compounds will inhibit the process. And along with ACE2 receptor, the proposed compounds will also inhibit the TMPRSS2, which also prevent the viral entry. So this is our second target. Okay, and the main target is the chymotrypsin like protease, or main protease, because it cleaves the polypeptides into small proteins. And our fourth target is RNA dependent RNA polymerase. So along with this, we have taken our immuno related target, which is the IL-6, because we know very clear during these there is so many systematic studies, and we have the strong evidences. So these patients know, when we check this markers, especially this pro-inflammatory markers, the IL-6 levels are very high. So that prompted us to take this IL-6 also as a prime target. So these are all our 5 targets we have selected for our study. Yeah, so as I told you, the main protease, so it is also known as 3CL Pro, that is 3 chymotrypsin like protease, which is regarded as responsible for the cleavage of viral peptides into functional units for viral replication and packaging within the host cells. Along with this, we have taken RNA dependent RNA polymerase, which is a very important enzyme which is responsible for viral RNA synthesis. And the main entry, that is angiotensin converting enzyme 2, that is ACE2 receptor. Then we have taken the TMPRSS2. At last, we have taken the as I told you, the cytokine storm. It will happen for many COVID patients. So the IL-6, it plays the crucial role in this disease. So we have taken these 5 targets. Okay. Now let us move to our work. So as I told you, we have taken 132 compounds. Okay, so these 132 compounds, just like that, we have not taken, which have been already reported for this SARS-CoV and MERS. Okay, so those 132 compounds, first of all, we have docked with this main protease. Okay, using lipdock protocol of the auxiliaries, DS. Okay, as I told you, they have supported us with the COVID evaluation license. Okay, yeah. Then from this, we have taken top 10 hits. Okay, based on the lopinavir, based on the known inhibitor lopinavir. Then again, further, this top 10 hits have been selected for ADMET screening. Then we have selected one particular compound. The particular compound, we have designed some analogs. The semi-synthetic analogs have been designed for a particular compound. Then for that design compound, we did the study with again all these 5 enzymes: MPRO, IL-6, RDRP, ACE2, then TMPRSS2. Yeah. So the first we did with structure-based visual screening using LibDoc. So we have used two modules of this DS. One is LibDoc. Then again we have taken the CDocker module. So we all know very clear it is a rigid-based docking program, whereas the CDocker is induced fit docking program. So this LibDoc, it calculates hotspots for the protein using a grid placed into the binding site and using polar and apolar and polar probes. So here we have taken the crystal structure of Empros, that is 1.7 angstrom. The PDB ID is 7BQ1. Then we have done the standard protocol, that is how it has the standard protein has to be prepared by removing crystal water and other heteroatoms except N3. N3 is the co-crystal which is present in the 7BQY, using addition of hydrogen, protonation, ionization and energy minimization. So the charm force field has been used and the minimization have been performed for 2000 steps with RMS gradient tolerance of 0.1. Then the prepared protein was used to define the binding site from the edit binding site option of the protocol. Then the active sites for docking were generated. Then visual screening was carried out by docking all the prepared ligands at the defined active site using LibDoc. So based on the LibDoc scores, the 132 compounds have been ranked. Okay, so this is our first step. We did with the 7BQI, that is Empro. All the compounds were ranked according to their LibDoc score. Then after that, again we did the top 10 compounds have been selected, and those compounds have been again docked using the CDocker module of the Discovery Studio for further molecular docking study. So CDocker is an implementation of a charm-based docking tool. The receptor is held rigid while the ligands were allowed to flex during the docking process. So here we have taken two parameters. One is CDocker energy, other one is CDocker interaction energy. Okay, so same thing here also we have used the protocol. The crystal water molecules were generally removed. Then the structure was completed. If any missing loops are there, it has been inserted by standardizing the names of the atom. Then the titrable residues were protonated. Then the potential energy, van der Waals energy, electrostatic energy and RMS gradient of the complexes were checked before and after protein minimization. Then here also the charm force field was used for energy minimization. The binding site pairs of all the proteins were defined as the regions that come within the radius of 15 angstrom from the geometric centroid of the native ligands. The binding site pairs of all the proteins were defined by the structural alignment. Okay. Then during the docking process, the ligands were allowed to bind to the residues within the binding site space. So we all know very clear we have to validate the docking process. So for that, the co-crystal has been taken, and once again it has been redocked into the crystal structure of all proteins. So here all proteins in the sense all the targets, like starting with Empro, RDRP, then AS2, TMPRSS2 as well as IL6. Okay, so all the validation has been done, that is the co-crystal has been taken. Once again it is redocked into the crystal structure. The RMSD between the docked pose and the crystal structure of the complex was less than 1 angstrom, indicating the CDocker module was highly reliable for reproducing the experimentally observed binding mode of all proteins. As I told you, we have calculated the CDocker interaction energy and CDocker energies for this protocol. Yeah. So this is our results. So first of all, we have taken the Empro, that is 7BQY. Then the prepared protein was validated by using the Ramachandran plot. Then the lipdog score of top 10 constituents among the 132 compounds. Okay, so these are all the 10 compounds which are having the lipdog score similar or more or less the standard remdesivir. Yeah, so here cinnamontanine B1. So it is the top scorer. It was the lipdog score was higher than the score obtained for remdesivir. And this 10 compounds again, once again, it has been run under the C-Docker module of the DS. So the C-Docker energy of gallocatechin gallate and cinnamontanine B1 were lower than the reference remdesivir. So indicating that these two compounds elicit a higher binding affinity with MPROS. Okay. So now from 132 compounds, we have narrowed down to 10. From 10, again, we have come down to two compounds which are gallocatechin gallate and cinnamontanine. Both are polyphenols. Further, these results are validated, or these results are, yeah. So these results, once again, we have checked by the amino acid interactions, and the amino acid interaction, especially hydrogen bond, pi-pi stacking and hydrophobic interactions have been checked with the standard remdesivir. So you can see here, this is the cinnamontanine B1 interaction, and this is the second one is gallocatechin gallate, and these interactions are compared with the standard remdesivir. Yeah. So the ligand binding analysis. So to study the ligand binding mechanisms, the top 6 ranked compounds were docked into the 3D structure of MPROS by using the C-Docker module of DS. The C-Docker energy as an estimation of molecular complex binding affinity was used in this study. So, as I told you, these two compounds, gallocatechin gallate and cinnamontanine B1, were lower than the reference. Yeah, so the remdesivir indicating that these two compounds elicit a higher binding affinity with MPROS. So, the increased binding affinities are further supported by the comparative analysis of receptor ligand interactions with the active amino acids of residues of MPROS. So the major non-bonded interactions, we know very clear there are two types of interactions, bonding interactions and non-bonding interactions. So the non-bonding interactions such as hydrophilic, hydrophobic and pi-pi stack interactions. So all these interactions are compared with the standard remdesivir. So here, interestingly, we found that the hydrogen bond interactions are more than that of the standard. For instance, that remdesivir showed only 5 interactions, whereas this gallocatechin gallate and cinnamontanine formed 8 and 7 pairs of the interactions respectively. Not only that, both of the compounds have shown the strong pi-pi interactions also, because they are having the benzenoid nucleus in the structure, both gallocatechin gallate and cinnamontanine. Yeah. Then after that, we have chosen these two compounds for our next study, gallocatechin and cinnamontanine. So, we know very clear the Lipinski rule of for orally active compounds, it should obey the Lipinski rule of 5. So, it is an important parameter of oral drug candidate. So, when we analyze the rule of 5 of two compounds, gallocatechin gallate and cinnamontanine, violations of the gallocatechin were 3 and 2 respectively. So it's a well-known fact that beyond the violation of 2 is questionable. Based on these results, we decided to proceed with gallocatechin further. We have excluded the cinnamontanine. Okay, and not only that, again we have done a simple small modification. Chemical modification of naturally derived compounds may be required to increase the potency. So working on these principles, we designed 6 analogs of gallocatechin. So these are the 6 analogs of the gallocatechin C. Yeah. So here, this as we know, this gallocatechin, it contains 4 ring systems, especially A, C, and D are aromatic. So to alter the hydrogen bond donor and acceptor and to increase the hydrophobicity, the hydroxy groups of ring A and C of gallocatechin are removed in a sequential manner to derive the analogs. So we derived 6 analogs starting with 1, 2, 3, 4, 5, 6, 7. So here these 2 hydroxy and 3 hydroxy groups are removed in sequential manner, and we designed these 6 analogs. And after that, these six analogs have extensively done that we have gone for the docking studies by using the C docker module of the DS. So you can see the results over here. So all the compounds are all the results, the C docker interaction energy results are compared with the standards. Okay, like lopinavir for Empro, ramipril for ACE2, camostat for TMPRSS2, and favipravir for RDRP. And for IL-6, we know very clear there are monoclonal antibodies or the known inhibitor. So we have taken the co-crystal tartaric acid as the standard for comparing the C docker interaction energies. Okay, yeah. So, and after that, the results indicating the compounds may possess COVID-19 combating activity through the inhibition of these proteins. Among all, the compounds GC3 and compound GC6 shows significant activity towards all the enzyme. So these are all the PDB IDs of all the proteins what we have taken for our study. So we came to know that compared to this MPRO and TMPRSS2, these ACE2, IL-6 and RDRP, all these 6 compounds are showing they are comparatively they are showing very good interaction energies. RDRP, they are showing a very good results compared to the standard favipravir. And further we have to validate with molecular dynamic study. So we have taken the GC6 compound for the molecular dynamics study. So in computational drug discovery, this MD is a technique that sheds light on the allosteric binding site of the protein and confirmation of the ligand protein complex. Also, it can simulate the conditions that are hard to perform in wet experiments. So here the GC6 was subjected to MD simulations, this one against RDRP. So the initial confirmations were acquired from the molecular docking experiments by C docker. It can be seen from figure 3A, that is the compound G6 retained its binding affinity and was firmly bound to its respective binding site. So the RMSD curves of the receptor structures from each complex and the potential energy profiles of each complex are shown in figure B, 3B and figure C. Not only that, like the trajectories of the complexes reached equilibrium after 10 nanoseconds. RMSD and the potential energy of the complexes get stabilized with time. The hydrogen bond distances formed between GC6 and RDRP were within a range of around between 3 to 2.3 angstrom. Besides the GC6, it formed 3 pairs of hydrogen bonds with water molecules. After molecular dynamic simulations, we can see the results. Okay, yeah. So these hydrogen bonds might contribute to the stability of the complex. Then the RMSD values of each receptor ligand complex were plotted to elucidate the effect of the compounds on the structural stability and integrity. So from this plot, it showed sharp increase in RMSD up to 2 nanoseconds. Later, deviations are optimized up to 80 nanoseconds. Then it increased slightly to 0.75 angstrom. Further, the complex attained equilibrium till the end of the dynamics. Yeah. So, what may be the reasons for this? So, we know very clear these polyphenols, especially like those, it will be having the aromatic with so many hydroxy groups. There are so many literatures you can see all these, especially green tea. Green tea is has been it is showing very good activity against SARS-CoV-2 due to this catechins, epigallocatechins, all these things. Okay, yeah. So it was well known that hydrophobicity plays the major role in predicting the binding affinity towards SARS-CoV-1. So the several natural sources from flora and fauna have been tested in terms of anti-SARS-CoV-1 activity and used as a scaffold in drug development since its outbreak in 2003. So, as I told in the beginning, it may include flavones, flavonols, fatty acids, tannins, terpenes, and alkaloids. Yeah, so there is a literature, it says that this SARS-CoV-1 inhibition requires chemical structures containing a hydrophobic aromatic ring, hydroxyl groups, and carbohydrate moieties. It was well evidenced by our results that the identified compound has hydrophobic and multiple hydroxy groups. Okay, so it is very well correlating to our results. Okay, and what I can say that is all these results, they are mainly, they are targeting to mpro only, main protease only. So there are limited studies where these polyphenols are effective against all these targets. Okay, so yeah, so although the importance of MPROS has been established as a prime target for COVID 19, there are targets like RDRP and ACE2, TMPRSS2, and IL6 have not yet been explored. So, there were also limited studies for these polyphenols and catechins. As I told, this poly targeting is also a reasonable approach to avoid issues related to resistance and toxicity. So, keeping all these views, we wanted to study the gallocatechin and its analogs against these multiple targets. So, interestingly, we observed the best binding affinities of gallocatechin and its analogs against RDRP, ACE2, TMPRSS2, and IL. Okay, yeah. So, though this GC, that is gallocatechin, it is a potential hit against these multiple targets. It fails to satisfy this rule of five. So, it's a very basic rule for oral drug candidates. So, in this view, we design analogs of GC where the OH groups have been modified to satisfy and all were within the range. All the design analogs have shown a comparable binding affinity with GC itself. However, the compound GC6 was identified the topmost hit. So, by considering the binding energies, amino acid residue interactions, and molecular dynamic studies, we propose the following pharmacophoric query for having target inhibitory potentials of SARS-CoV-2. The ring aromatics should be more than 3. The hydrogen bond acceptors is equal or more than 8. Hydrogen bond donor should equal or more than 5. Sorry, less than 5. So the conclusion, coming to the conclusion, the rapidly spreading outbreak of COVID-19 has challenged the healthcare sector of the world in past the 2 years. The several investigations have demonstrated that a variety of plant secondary metabolites, especially polyphenolics, have been reported for anti-SARS-CoV-1 and 2 activities since 2002. So we aimed to identify any single molecule that can target all important targets involved in SARS-CoV-2. So we carried out the systematic structure-based in silico study and found these compounds 2 or 3R 2,3,4 dihydroxyphenyl, chromanyl 3,4,5 trihydroxybenzoate as a multiple targeting inhibitor. However, further studies are warranted for the validation of these compounds using in vitro and in vivo models to pave a way for these compounds in anti-COVID-19 drug discovery. Yeah, so the work has been published in RSC Advances in the year of 2021, April, and we got hot articles also, selected and featured in hot articles. And he's my collaborator, Dr. Suresh K. Mohan Kumar. He's in UK, Swansea University. And yeah, I'll be concluding my talk with acknowledgement. I'll be grateful uh, for my leadership and management of JSS College of Pharmacy UT, as well as JSS Academy of Higher Education and Research and Altum Technologies Private Limited for providing an evaluation license for COVID-19 research. Without that, it wouldn't have come to this level. Especially, I'll be thanking to Ms. Divya Sanmugarajan, Senior Application Scientist. And a big thanks to you all. Thank you.
Thank you so much, Ruby ma'am. It's a great and informative session, and I hope everyone enjoyed the session. Yeah, Ruby ma'am. Thank you so much.
Thank you, ma'am. Thank you so much.
It's a great session, ma'am, and you explained very well in detailed manner, and each and every point of the things you are shown very in atomic level. I could say that atomic level you have explained that in very detail. I think still we have a few questions or in the live chat, ma'am. Shall we go ahead, ma'am?
Yeah, please.
Okay.
Yes,
Yeah,
ma'am.
ma'am.
Yes. I can't see here. Yeah.
Yeah, ma'am. I think it is not.
You can. Yeah.
Yeah, ma'am. I think a few. I am picking the few questions. First of all, what is the importance of polyphenols for the drug discovery process? Importance of polyphenols in drug discovery.
So, as we all know, usually polyphenols in drug discovery. Yeah. So we know very clear. Normally, we consider the score first. It may be the binding energy, or the next part is normally we compare the two-dimensional interactions with the co-crystal or with the standard. So these polyphenols, it will provide the hydrogen bond interaction as well as the pi stack interactions. So that's the major reason these polyphenols are normally in the drug discovery process. Yeah.
Okay, ma'am. Thank you, ma'am. Thank you so much. And then, can we perform ADMET studies in the software?
Yeah, definitely. Actually, I have excluded that part. We have done the ADME studies, or we have done the ADME. First, we did with the libdol. After that, we did. Yeah, we can do the ADME studies also and toxicity studies also. There is a TopCat module in the software. You can perform the toxicity studies also. I think Divya madam will be explaining more in this aspect.
Yeah, definitely. I would like to add few words with Dr. Juby ma'am's extension. See, this software, BioVR Discovery Studio, has a more potential to predict ADMET properties and then TopCat properties of various animal models. All these models are built by using a machine learning algorithms and various data sets based on FDA and NPP will add a more significance to the software. So as we have seen that it is reliable and people are doing a lots of research on the software and many, I could say that it's a very user-friendly software that everyone can use. And we have a final question with you, ma'am. And then, okay. There is a question from Dr. Suresh, sir. Can we inhibit enzymes responsible for the release of virus using this kind of a study? I will come again once again, ma'am.
Ma'am,
can we?
Yeah, ma'am.
Can we? I could reframe and say, can we study the inhibition of enzymes responsible for this release of this virus? That means you are telling about MC prodrome. There will be a enzyme site will be there. can we see the incubation? That is what, if I'm right.
Yeah, I think, ma'am, it is silico level can we do, or in vitro enzyme binding assay, SAR is mentioning, ma'am.
Yeah, I think if it is there, it could be like it should be from an in silico only, as we are discussing this one. So, can we study the inhibition of enzyme responsible for release?
I think,
I think it is.
maybe,
It,
sir, I am not very much
of
sure
course,
about that.
yeah, I will just add
Yeah,
with
in
you, ma'am.
vitro level we can, yeah, in vitro level we can do the binding studies. Yeah, please, ma'am.
Yes, ma'am. So we can see the inhibition of enzymes because enzymes will have a catalytic sites. So if you know the catalytic sites of this enzyme, especially a triad or diad or whatever is it, you can point out those amino acids and you can see the incubation and binding activity. However, when you want to extend to studies responsible from an endocytic vacuole, that is inside the cell cycling process, which has to perform only in the in vitro. And I think there is also one more
Yes.
question. No. Okay, polyphenols can be studied. I think we have asked it. Admit we asked it. And then what is the advantage of this lip dock over the autodock? I think uh, I will just take up this question and then see. Lip dock is basically,
Yeah.
yeah, I
So
could say that.
what I can say, yeah, please.
Yes, ma'am, please, ma'am, please, ma'am. Once you finish, I will continue. Lip dock over autodock, ma'am. Please, ma'am.
Yeah. So, yeah, autodoc also, when we have 100 compounds, or we have a huge library of compounds, so initially we can start with rather than directly going to the autodoc. So you can narrow down the compounds. You can select the compounds from the lib, then you can go for autodoc, because it is like, it's a hotspot program. So it will select the particular moiety only. So you can fine tune the molecules, then you can go for autodoc. I think madam will be explaining more.
Yeah, thank you, ma'am. Thank you so much. See, in the autodoc and all, whatever is it, we'll be getting only with a few library of compounds. If you have more than lakhs of compounds which need to be screened and you want to understand its interaction in a one show, I think this software of which containing the docking program LibDoc will help you to screen initially based on the hotspot, which basically uses a Ludi algorithm and then will shows along with the hotspot docking, you can able to see the compounds very well, as Juby ma'am said. I think so. It's a very great session, ma'am, and we got a very great feedback. And anything, and if you have peoples, you have watchers, YouTube watchers. If you have any queries, feel free to put your questions over there in the live chat. Okay. So we have no questions as of now, ma'am. I think we are done with it, and it is a great thing and everything. It's a very informative session, and people who is working on a natural products have a will definitely has to see this, because how the natural products based will help you to have an better drug discovery perspective. And even whenever the pandemic arises, I think we should not get afraid to go back. We have already solutions in your hand. We can use these kinds of natural products, and we can also, you made a great modifications in that. It's really appreciable. And then how well you are planned and you have work it out to identify a new lead compound for an targeting MP Pro. And we wish you all the best for your future endeavors, ma'am. Like this, we want you to succeed the many things which using this kind of a software and to come out with an best results, ma'am. We wish you all the best for you, ma'am. And thank you so much for
Yeah, thank
being
you,
here
ma'am. Yeah.
as the
Thank you
invited.
once again.
Thank
Yeah.
you so much, ma'am.
Yeah. Once thank your you and your company's generosity to provide me evaluation license. I hope that we can do this work. Once again, I want to thank your company. Thank you so much, ma'am. Yeah.
Yeah.
And
Thank
thank
you,
you
ma'am.
all.